Our DNA is a complex and fascinating blueprint of life. It dictates our traits, from eye color to our likelihood of getting certain diseases. For decades, scientists mainly focused on the small percentage of DNA that codes for proteins, the building blocks of our bodies. The rest, the overwhelming majority, was dismissed as junk DNA, considered useless and unimportant. This seemingly barren genetic wasteland, however, has recently become a hotbed of groundbreaking discoveries. Scientists are discovering that junk DNA is not useless at all. It plays a key role in regulating gene expression, influencing everything from embryonic development to disease progression. The historical view of junk DNA as useless reflects the ever-evolving nature of scientific knowledge. As we delve deeper into the intricacies of the genome, we begin to appreciate the true complexity and elegance of our genetic makeup. The so-called junk DNA, once relegated to the shadows, now emerges as a key player in the symphony of life. In the 1970s, the term junk DNA was coined by geneticist Susumu Ono. Ono, known for his groundbreaking work on chromosome evolution, observed that a large part of the genome does not code for proteins. Given the limited understanding of DNA at the time, it seemed logical to assume that these non-coding regions were simply evolutionary leftovers, remnants of past genetic experiments that served no present purpose. This view was further reinforced by the discovery of pseudogenes, stretches of DNA that resemble functional genes but lack the ability to produce proteins. These fossil genes were seen as further evidence of the wasteful nature of the genome, cluttered with the remnants of evolution. However, as with many scientific paradigms, the junk DNA hypothesis was destined for revision. With the advent of new technologies and a deeper understanding of gene regulation, scientists began to uncover hidden layers of complexity within these supposedly useless regions. The term junk DNA, while catchy and provocative, proved to be a gross oversimplification of a much more nuanced genetic landscape. Far from being inert fillers, non-coding DNA sequences, now often referred to as dark matter DNA, are teeming with activity. They contain a plethora of regulatory elements such as enhancers and promoters, which act like switches, turning genes on or off and controlling their activity levels. These regulatory elements are crucial for the precise orchestration of gene expression, ensuring that the right genes are activated at the right time and in the right cells. One of the most intriguing discoveries is the role of junk DNA in the intricate dance of embryonic development. Specific non-coding sequences act as master regulators, directing the differentiation of stem cells into the specialized cell types that make up the diverse tissues and organs of a developing embryo. Disruptions in these regulatory elements can have severe consequences, leading to developmental abnormalities and diseases. Moreover, junk DNA has been linked to a wide range of diseases including cancer, heart disease, and autoimmune disorders. Mutations in non-coding regions can disrupt the delicate balance of gene regulation, leading to uncontrolled cell growth, inflammation, and other hallmarks of disease. Understanding the role of junk DNA in disease is crucial for the development of new diagnostic tools, targeted therapies, and personalized medicine approaches. Perhaps one of the most unexpected discoveries is the presence of ancient viral DNA within our genome. Over millions of years of evolution, viruses have inserted their genetic material into the DNA of our ancestors, leaving behind remnants of their past invasions. While some of these viral sequences are inactive, others have been co-opted by our cells for beneficial purposes. For instance, some viral elements have been repurposed into essential components of our immune system. They play a role in recognizing and combating invading pathogens, providing a crucial line of defense against infections. This remarkable adaptation highlights the dynamic and often collaborative relationship between viruses and their hosts. The presence of viral DNA in our genome is a testament to the ongoing evolutionary arms race between viruses and their hosts. Viruses, with their ability to rapidly mutate and adapt, have driven the evolution of our immune system, shaping our genetic makeup and influencing our susceptibility to disease. Understanding the interplay between viral DNA and our genome is crucial for the development of new antiviral therapies and vaccines.